Hi everyone, welcome to my channel. In this video, we shall try to learn about the concept of enzymes. If you consider a living tissue, the living tissues are able to bring about hundreds of chemical reactions per every minute of their life to obtain energy or to synthesize the protoplasmic and other structural materials or to absorb from the external environment that are required for the life, or else the unwanted materials that are present within the life, they are supposed to be excreted or eliminated out of the life system. In order to carry out all these things, the tissues will perform various chemical reactions. Say for example, if you consider the hydrolysis of proteins into amino acids. For the process of hydrolysis of proteins into amino acids, if you want to conduct the same outside the cell, they are supposed to be heated for several hours in presence of chemicals such as either acids or alkalis. So it's more laborious and more time consuming process, that is the hydrolysis of proteins into amino acids. As well, for the synthesis of proteins as well as some other complex compounds, we generally use the chemicals as a catalytic material so as to increase the rate of the reactions. But usage of chemicals always for the synthesis of these complex compounds and in order to perform such a kind of reactions is not possible at all the time. But such a kind of reactions were actually occurs within the cell more fastly even at the very low temperature of 37 degrees Celsius and at a moderate pH of 7.4 itself. How come it is all possible? This thing is mainly possible because certain proteinish materials will act as a catalyst in presence of some non-proteinic components, and that carries many biochemical reactions in a much faster rate so as to yield the products. And such proteinaceous materials which are acting as a catalyst in biological reactions are simply called as enzymes. So we can simply define the term enzymes as the enzymes are biocatalysts that alter the reaction rates of biological system, and that's the simplest definition we can given for the term enzyme. Okay, but if you come to the whole and soul nature of these enzymes, we can say the enzymes are CTPS in nature. So it is the mnemonic we can remember to judge the nature of the enzyme. So that is, the enzyme is colloidal in nature. Enzyme is colloidal and it is temperature labile. That means it reactive towards the temperature, and it is proteinaceous. That means enzymes, majority of enzymes are proteins except ribosome. All the remaining enzymes are proteins in nature. This ribosome acts as a catalyst, and catalytic activity is due to the presence of RNA molecule. And S is account for specificity. That means the enzymes are highly specific in nature. That means each and every enzyme will act towards only one type of material or only one type of reactions. That means each enzyme, if you consider an enzyme, it is not possible to use the same enzyme to carry out the reactions of all the types. Each enzyme is specific towards a particular type of reactions or particular substrate that we shall look over in the next coming topics. So here you should remember this one. The enzymes are CTPS in nature. That means they are colloidal, temperature labile, proteinaceous, and they are highly specific in nature. And if you come to the case of how actually an enzyme will work in case of a biochemical reactions or in normal reactions, for that let us consider one simple example. So if you consider a hill and the train has to climb this hill so as to reach this target point. So here the train has to climb this hill and it has to come over to this target point. And in order to climb this hill, this train is supposed to require more amount of energy and it needs more time also. Right? Suppose instead of this direction, if we have created a tunnel over here, then the train can easily move through this tunnel and can reach the target point. Right? So when train is moving in this tunnel, it needs less amount of energy compared to this direction, and less time is required, less energy is required when train is moving in through this tunnel instead of climbing the hill. In the same way, we can say over here that the train can reach the target position through this alternate path instead of climbing the train. Understood? In the same way, an enzyme will create an alternate path for the reaction. An enzyme will create an alternate path for reaction so as to convert the substrate molecule, that means the substance which is undergoing transformation. In the field of biochemistry, we used to call the reactant molecules as substrate molecule. That means the substance which is undergoing transformation. So an enzyme will create an alternate path for the reaction, so that the enzyme will create an alternate path for the reaction so as to convert substrate into product. And as we said, by the usage of enzyme, the time required for the conversion of substrate into product will be reducing, as well the energy needed for the reaction as to takes place will also be reduced. And because of all these things, when enzyme is used, it catalyzes the biochemical reaction, and that also produce a good yield of the reaction, mean more amount of product will be obtained, and that is the capability of an enzyme. If you come to the characteristic properties of this enzyme materials, so enzymes are highly active materials, and each and every enzyme is having capability so as to convert roughly around 100 to 1000 substrate molecules into products. So this is the least capability of the enzyme material, but each and every enzyme can convert more amount of reactant molecules, that means substrate molecules, into product also. But in general, we can say each and every enzyme molecule can easily convert 100 to 1000 substrate molecules, 100 to 1000 substrate molecules into product per second of time. That is the capability of an enzyme. So in this way, if you consider one enzyme molecule, and if you added that enzyme molecule for a reaction mixture or for a reaction system so as to convert the substrate molecules into product, the number of substrate molecules, if an enzyme molecule is added to a reaction system, the number of substrate molecules that can be converted into product by one enzyme molecule per second. So the number of substrate molecules that can be converted into product by one enzyme molecule per 1 second is called as turnover number of the enzyme. Turnover number of that particular enzyme molecule. So if you consider almost all the enzymes that are available in the field of biochemistry, so among all the enzyme molecule, the carbonic anhydrase is the enzyme which is having the highest turnover number of 360 lakhs. 360 lakhs is the turnover number of that particular enzyme, that is carbonic anhydrase. So that is the most important concept of the enzyme, that is turnover number. As we said, the enzymes are proteinaceous in nature, and these enzyme proteins will be always in association with some non-protein components, and those non-protein components will be called as prosthetic groups. And that prosthetic group which is in association with the enzyme protein may be either a metal ion or maybe a low molecular weight organic materials or organic complex. The prosthetic group which is in association with the enzyme protein can be a metal ion or it can be a low molecular weight organic molecules. Okay, and presence of prosthetic group is very much required for a enzyme protein so as to make it active, because only the protein content is not that much effective or not active in order to conduct the catalytic activity in case of a particular chemical reaction, strictly speaking a biochemical reaction. When that enzyme protein is in association with a prosthetic group, such as it may be a metal ion or a low molecular weight organic complex, in particularly the low molecular weight organic compound, the protein, strictly speaking, the enzyme protein will become more active and that performs more catalytic activity. That means presence of a metal ion or a low molecular weight organic complex, that is a prosthetic group along with enzyme protein, will make it active, and that increases the catalytic efficiency of that enzyme protein. Okay, and the metal ion, that is prosthetic group that can be present along with the enzyme protein. We can give some simple examples for the metal ions which are acting as a prosthetic group with enzyme protein. Some examples may be Fe2 plus ion, Mn2 plus ion, or they can be zinc ion, or it can be cobalt 3 plus ion, etc. Like this we can give many examples. And the low molecular weight organic complex, anything it can be, any organic complex may present along with the enzyme protein. And the metal ion prosthetic group which is present along with the enzyme protein will be called as cofactors. And the low molecular weight organic complex which is present along with the enzyme protein will be called as coenzyme. Coenzyme. Both of these things, that is cofactors as well as coenzymes, will increases the catalytic activity of the enzyme. And as we said, the enzymes are consisting of protein as well as non-protein component, right? And the protein part of the enzyme, the protein part of enzyme which is not active to conduct the catalytic activity, right? So the protein part of this enzyme is simply called as apoenzyme. The protein part of the enzyme is called apoenzyme, and the non-protein part of the enzyme, as we already said, in case of enzymes compared to the activators, most of the time we will be having a coenzyme, that is low molecular weight organic compound, which is the prosthetic group, right? So that non-protein part of the enzymes will be called as coenzyme. This apoenzyme will be inactive in inactive form. Once the coenzyme joins to the apoenzyme, that makes a conjugated complex, and that conjugated complex formed by combination of this apoenzyme and coenzyme will be active, and that will perform the catalytic activity. So that the conjugated active form, the conjugated active form of apoenzyme and coenzyme. The conjugated active form of apoenzyme and coenzyme will be simply pronounced as holoenzyme, and this holoenzyme is actually the active form of the enzyme, and this will actually conducts the catalytic, or this will actually shows the great catalytic activity, and that we shall see how these things will happen. Here you can see the apoenzyme, which is the protein portion, which is inactive to show the catalytic activity. But when it comes in contact with a cofactor or a coenzyme, which is a non-protein, that activates the apoenzyme so as to produce the holoenzyme, which is the active form that receives the substrate molecule and makes the sufficient changes or biological reaction that is required, and with that it produce the product material, and thereby the enzyme will get regenerate, and the regenerated enzyme will again receive the substrate molecule so as to produce the product. Now we shall move on to the classification of enzymes. So based on the recommendations given by the Enzyme Commission, all the available enzymes in the field of biochemistry have been classified into 6 major classes. And in order to remember the 6 major classes, we can have one small mnemonic as OTHLIL. You can remember this mnemonic. So like this, you can frame a sentence to remember this mnemonic: on the high level interior look. Just you can remember this: OTHLIL. So total 6 categories of enzymes. And now we shall look over to the each and every type. The first class, that is O, accounts for Oxido Reductase, and the second letter T accounts for Transferases, and third one H accounts for Hydrolases, and fourth one is Lyases, I Isomerases, L Ligases. I repeat: Oxido Reductases, Transferases, Hydrolases, Lyases, Isomerases, Ligases. So totally 6 major classes of enzymes. And now we shall go on to the each and every type, and now we shall look over the exact nature of each and every type of enzymes with suitable exemplifications. So in that, the very first one is Oxido Reductases. Oxido Reductases. The name itself telling you these are the enzymes which catalyzes the process of oxidation and reduction. That means the substrate molecule can be subjected to the process of oxidation, or they may catalyze the reduction of a reaction, reduction of the substrate molecule. And how the oxidation or reduction of the substrate molecule will occur? Simply by transfer of the hydrogen or by the transfer of electrons. So oxido reductases are the enzymes that catalyzes oxidation or reduction of substrate by transfer of either hydrogen or by electrons, right? So we shall give one simple example for this oxido reductase type of enzymes. So if you consider the lactate molecule, and the lactate molecule is supposed to be converted into pyruvate. And in order to convert lactate into pyruvate, we need to remove one molecule of hydrogen from lactate. So we need a receptor molecule, and the receptor we use is NAD. NAD accounts for nicotinamide adenine dinucleotide. NAD, that is nicotinamide adenine dinucleotide. That's the full form of NAD. So this is the receptor molecule over here that receives one hydrogen molecule from the lactate molecule so as to produce pyruvate. But for this purpose we need a catalyst. That means we need an enzyme. So substrate molecule is lactate, and what the thing is going on? Removal of hydrogen, transfer of hydrogen from lactate to NAD. So it means the enzyme is supposed to remove the hydrogen from lactate. That's what we call as dehydrogenation. So the catalyst which is acting over here is lactate dehydrogenase is the enzyme. So lactate losing hydrogen molecule so as to convert into pyruvate, and the lost hydrogen will be received by this NAD, that is nicotinamide adenine dinucleotide, and among the hydrogen molecule which is coming out of the lactate, this NAD will pick one hydrogen so as to produce the reduced form of it, that is NADH, and one hydrogen will comes out in the form of H+. So here you can observe the hydrogen is removed. That means oxidation of the lactate have been occurred. And similarly, we can take one more example. Let us consider L-glutamate. L-glutamate, and this L-glutamate is reversibly changes into alpha ketoglutarate. Alpha ketoglutarate, and here the receptor molecule used is NADP. That means nicotinamide adenine dinucleotide phosphate. P accounts for phosphate. And here also L-glutamate is converting into alpha ketoglutarate, and the enzyme we use is glutamate, right? It is the substrate molecule. So glutamate dehydrogenase is the enzyme we use over here. And in presence of glutamate dehydrogenase enzyme, L-glutamate will lose one hydrogen molecule to NADP, and this will reduce us to give NADPH, and one H+ will come out, and that along with that there will be elimination of NH4+ also. So by eliminating NH4+ and one hydrogen, L-glutamate is converting into alpha ketoglutarate. Here also we are observing elimination of hydrogen, so that oxidation reduction have been taken place. So this is one set of enzymes that performs the oxidation or reduction, that what we call as oxido reductases. Now the second type of, or the second category of enzymes are called transferases. Transferases, the name itself telling you, they are the enzymes that catalyzes the transfer of components, and what they transfer, that we are supposed to understand, that we are supposed to learn. And these are transferases, or the enzymes that transfer the groups from substrate molecules to acceptor molecules. And here the transfer of groups will occur and the group doesn't include hydrogen, and they transfer the groups other than hydrogen into acceptor, and the acceptor will be anything other than water molecule. Okay, so transferases are the enzymes that catalyzes the transfer of groups, from substrate to acceptor. These are the enzymes which catalyzes the transfer of groups from substrate to acceptor, and the groups can be anything that will be other than hydrogen. So except hydrogen, any group can be transferred with the transferase, and the acceptor will be anything and that will be other than water. And these are the conditions in case of transferases. And for this, we shall give one simple example for transferases. If you consider say glucose, and glucose is need to be converted into glucose 6-phosphate. Glucose 6-phosphate. Okay, so here the enzyme used will be hexokinase. In presence of the enzyme hexokinase, ATP will take part in the reaction that transfers one phosphate group to the glucose to the 6th position so as to produce glucose 6-phosphate, and ATP will get converted into ADP. That means adenosine triphosphate will convert into adenosine diphosphate. This is one of the simple example we can consider so as to say the transferase. Okay, and here ATP is transferring 1 unit of phosphate into glucose so as to produce glucose 6-phosphate. That means the transferase enzyme is, that is hexokinase, catalyzing transfer of groups from substrate molecule to acceptor molecule so as to produce a product, and the group will be other than hydrogen, that is here we are transferring the phosphate molecule, and that is other than water molecule, that is to glucose. That's the thing about transferase enzyme. Now the third category of enzyme molecule, they are called as hydrolases. That is the third category of the enzymes. The name itself telling you hydrolases. That means these are the enzymes that catalyzes the process of hydrolysis. That means they catalyzes the hydrolytic. We know that hydrolysis is a breakage of bonds in a molecule, and the hydrolysis in case of biomolecules, in case of biochemistry, these hydrolases enzymes will catalyze the hydrolytic cleavage of covalent bonds in biomolecules. And which are those covalent bonds, that also we need to understand. Those covalent bonds may be either peptide bonds, that's nothing but amide linkages that are present in case of peptides, or that covalent bond may be an ester link, which is present in esters, or in case of triglycerides, or the covalent bond may be an glycosidic bond, which is present in case of oligosaccharides and polysaccharides, glycosidic link or glycosidic bond, or it can be an anhydride bond. So wherever these bonds are present and whenever the breakage of these bonds are supposed to happen, these hydrolases enzymes will comes into picture, and in presence of water they break the bonds, break these bonds and produce the needy products. So that we can say hydrolases are the enzymes that catalyzes the hydrolytic cleavage of covalent bonds, and these are the type of covalent bonds which are going to break in presence of water under the influence of the enzymes hydrolases. So let us give some simple examples for hydrolase enzymes. If the hydrolysis of urea is supposed to happen reversibly, the hydrolysis of urea reversibly occur in presence of the enzyme urease, and the amide bond present in urea will get break so as to produce carbon dioxide, and it releases 2 molecules of ammonia. So this is a process that is catalyzing by the enzyme urease, that is it catalyzing breakage of bonds in urea in presence of water. And we can give one more example for this hydrolase enzyme. Say if you want to carry out the hydrolysis of beta galactosidase is supposed to be hydrolyzed H2O, and this also reversibly occurs, and the enzyme required is beta galactoside is the material, and in order to carry out the hydrolysis of beta galactoside we need beta galactosidase is the enzyme needed for performing hydrolysis of the material beta galactoside, which is the substrate molecule. When beta galactoside is hydrolyzed in presence of beta galactosidase, it results in the formation of beta galactose. Beta galactose will be formed, and along with beta galactose an alcohol will be formed. So here in both these cases you can observe there is a performance of hydrolysis of the substrate molecule in presence of the suitable enzymes. And that's about the third class of enzymes, that is hydrolases. The fourth category of enzymes are Lyases. Lyases are the fourth category of enzymes, and these are the enzymes that catalyzes the removal of groups from the substrate. Removal of groups from substrate. And here the thing is, you can observe in previous case also we said the removal of groups will be happened, or the transfer of groups will be happened in case of transferase enzyme. And here in a previous case we did the breaking of bonds in a substrate molecule through hydrolysis. But if you come to the case of lyases, these enzymes will catalyze, these enzymes also catalyzes the removal of groups from the substrate molecule. But the thing is, the mechanistic pathway that these lyases enzyme will follow is the one which is other than the mechanism of hydrolysis. So they don't undergo any kind of hydrolytic mechanistic pathway in order to remove the groups from the substrate molecule, and these lyases enzymes will not disturb the double and triple bonds. They just remove the other groups from the substrate molecule with the mechanism other than hydrolysis. hydrolysis. And that's about the concept of Lyases enzyme. So we shall take one simple example for Lyases enzyme, that is L-maltate. L-maltate can reversibly change into fumarate. And here there should occur elimination of some groups, right? So maltate will be converted into fumarate by the removal of water molecule. And for this purpose, the enzyme required is fumarase. Okay. And if you are having a material that is ketose 1-phosphate, and this ketose 1-phosphate is reversibly changes into an aldehyde molecule. Ketose is converting into aldehyde with the elimination of dihydroxyacetone. The elimination of dihydroxyacetone phosphate. Okay, so ketose 1-phosphate can be reversibly changes into an aldehyde by removing dihydroxyacetone phosphate. And for this purpose, the enzyme used will be Hydrolase is the enzyme that will be used for converting ketose 1-phosphate into aldehyde with the elimination of dihydroxyacetone phosphate. That means we can say lyases are the enzymes that catalyzes the removal of groups from the substance through the mechanism other than hydrolysis. And that's about the enzyme type lyases. Now, the fifth type of enzymes or fifth category of enzymes are isomerases. The name itself telling you isomerases. That means these are the enzymes that catalyzes the conversion of one isomer into another isomer. Strictly speaking, interconversion of isomers. And what are those type of isomers? Those isomers may be either optical isomers, or they may be geometrical isomers, or there may be positional isomers also. So we can say isomerases are the enzymes that catalyzes the interconversion of either optical isomers or geometrical isomers, or they may be positional isomers also. That's the nature of which one? Isomerases enzyme. So let us give one simple example for that. Let us consider glucose 6-phosphate. This is the substrate molecule, and this glucose 6-phosphate molecule, in presence of the enzyme phosphohexo, and the process carrying out is isomerase. Glucose is a 6-carbon containing material, so it is a hexo. And what is the reaction we are performing? Isomerization. So the name of the enzyme is phosphohexoisomerase. And here, when glucose 6-phosphate is reacted with phosphohexoisomerase, that results in the formation of fructose 6-phosphate. Okay, so one isomer have been converted into another isomer, that is glucose 6-phosphate is converted into fructose 6-phosphate. And let us consider one more example, that is what we can say. Let us consider D-glyceraldehyde 3-phosphate. D-glyceraldehyde 3-phosphate. And here we know that glyceraldehyde is a molecule which consisting of only 3 carbon atoms. So here we said phosphohexoisomerase, right? So here glucose is having 6 carbon atoms, so we use the term hexo. And here glyceraldehyde is having only 3 carbon atoms, so we can say triose, that is sugar consisting of 3 carbon atoms, triose phosphatase. Triose phosphatase is the enzyme in presence of which D-glyceraldehyde 3-phosphate will get converts into dihydroxyacetone phosphate. So here aldehyde is converted into ketone consisting of one hydroxy group also. That means aldehyde have been converted into ketol. So one isomer is changed into another isomer. That's what we are calling them as isomerase. Interconversion of one isomer into another isomer. Now we shall go with the last type of enzyme molecules, that is ligases. Okay, ligases or the enzymes, they actually catalyzes the coupling of the molecules, maybe whatever the molecules, and the coupling of molecules will occur in presence of a high energy components such as ATP, that is adenosine triphosphate. So what this ligases will do is they utilize the coupling of molecules in presence of ATP, as well they break down the high energy molecules or high energy components, high energy components into small energy ones. So that means ligases are the enzymes that catalyze the linking of the molecules through the process of coupling, and through that they break down the high energy components such as ATP molecules, GTP molecules, whatever it may be. So let us consider some simple examples so that we can understand what does the ligases molecules will do in actual sense. So for this, let us consider L-aspartate, and this L-aspartate will couple with ammonia in presence of ATP, that is adenosine triphosphate, and in presence of the enzyme such as okay, let us say in this way, L-aspartate will combine with or will couple with the NH3 in presence of ATP. It results in the formation of L-aspargine. And since aspartate upon coupling with NH3 we are getting aspargine, we call this as, the enzyme used over here is aspargine synthetase, since we are synthesizing the aspargine. So since with the usage of ligases enzyme, it is possible to synthesize the new biological components, the required ones. That's why the ligases enzymes are also called as synthetases. Ligases are also called synthetases. Since in this example we can observe from L-aspartate we are synthesizing L-aspargine, and here we said the high energy components are supposed to get breakdown, right? So here this ATP molecule will get breakdown so as to produce AMP, that is adenosine triphosphate, get breakdown to give adenosine monophosphate, and along with that it gives PPI, that means PPI in the sense pyrophosphate. That is 2 phosphate contents will be there. Here you can observe L-aspartate upon coupling with ammonia in presence of ATP, we are getting L-aspargine, through which ATP molecule got breakdown to give AMP with the production of pyrophosphate. phosphate. And shall consider one more example for this. Let us consider acetyl coenzyme A will couple with carbon dioxide in presence of adenosine triphosphate ATP. So in presence of the enzyme acetyl CoA carboxylase. Here you can observe the name of the enzyme acetyl CoA carboxylase. That means acetyl CoA getting carboxylated. That means the whole CO2 group will get add up. So when CO2 group got add up to acetyl coenzyme A, that results in the synthesis of malonyl coenzyme A, that is malonyl CoA, and this ATP will get breakdown to give ADP, that is adenosine diphosphate, with the expulsion of PI, that is PI is simply called as inorganic phosphate. So in this way, by using Lyases enzyme, we can synthesize the other materials that are required for the easy conduction of the reactions by the tissues. And that's about the overall classification of the enzyme molecules, which is suggested based on the recommendation of the Enzyme Commission. So in the next video, we shall learn about the active sites of the enzyme as well as the very particular property of the enzyme material, that is specificity. So if you like this video, please share it, do subscribe to my channel and press the bell icon so